Good day everyone, I am Angel Monteras from BSN1H. So today we will be discussing about glycolysis. What is glycolysis? Glycolysis is the metabolic pathway that converts glucose into pyruvate. The free energy released in this process is used to form the high-energy molecules adenosine triphosphate and reduce nicotinamide adenine dinucleotide. Glycolysis is a sequence of 10 reactions catalyzed by enzymes. Through a series of 10 enzymatic reactions. This process produces ATP along with other products such as NADH that can be used later to produce even more ATP for the cell. Let's watch these enzymes oxidize one glucose molecule into two pyruvate molecules. The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase, or it is called phosphoglucose isomerase. The third step is called phosphofructokinase or kinase, with magnesium as a cofactor changes fructose 6-phosphate into fructose 1,2,6 biphosphate. The enzyme aldolase splits fructose 1,2,6 biphosphate into two sugars that are isomerase of each other. These two sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate or GAP. The fifth step is all about triphosphate isomerase. The enzyme triphosphate isomerase rapidly interconverts the molecules dihydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate or GAP. Glyceraldehyde phosphate is used in next step of glycolysis. The step 6 is all about glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate or dehydrogenase or GAPDH and adds an inorganic phosphate to glyceraldehyde 3-phosphate, producing 1,2,3 biphosphogycerate kinase. Phosphoglycerate kinase transfers as a phosphate group for 1,3 biphosphogycerate to ADP to form ATP and 3-phosphoglycerate. The eighth step of glycolysis is all about phosphoglycerate mutase. The enzyme phosphoglycerate relocates the P from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphoglycerate. The step 9 is enolase. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenolpyruvic acid or PEP. The last step is pyruvate kinase. The enzyme pyruvate kinase transfers a P for phosphoenolpyruvate or PAP to ADP to form pyruvic acid, and ATP result in step 10. Steps 1 and 3 is equals to -2 ATP. Steps 7 and 10 is equals to +4 ATP. Net visible ATP produced is equals to 2. Immediately upon finishing glycolysis, the cell must continue respiration in either an aerobic or anaerobic direction. This choice is made based on the circumstances of the particular cell. A cell that can perform aerobic respiration and which finds itself in the presence of oxygen will continue onto the aerobic citric acid cycle in the mitochondria. If a cell able to perform aerobic respiration is in a situation where there is no oxygen, such as muscles under extreme exertion, it will move into a type of anaerobic respiration called homolactic fermentation. Some cells, such as yeast, are unable to carry out aerobic respiration and will automatically move into the type of anaerobic respiration called alcoholic fermentation.